Hello, everyone. Thank you for joining today's webinar, Benchling for Lab Automation, Harness the Full Potential of Your Lab Hardware. My name is Ankita Goswami, and I'm going to be your moderator for today. I'd like to introduce today's speakers, Rashi Tucker and June Crisaldo. Rashi is part of the product marketing team at Benchling, focused on the Benchling for Lab Automation product. Before joining Benchling, Rashi held various commercial and research positions at Forte Bio and Genentech, and has over 10 years of experience working with analytical and liquid handling instrumentation and platforms. June Crisaldo is a scientific solutions consultant at Benchling, working closely with our customers to identify the best ways to work with the Benchling platform, given their specific scientific use case. Before joining Benchling, Jun held various positions in the automated genomics space, most recently including a field applications scientist role for Eppendorf, where he consulted on automation and real-time detection platforms. Now, before we begin, I would like to go through some housekeeping items. Please feel free to submit questions at any time by clicking on the Q&A button at the bottom of the screen. We'll be sure to reserve time at the end of the webinar to answer them. And if we can't get through all the questions today, we will be sure to follow up with each of you over email. Additionally, as we go through, we will be polling the audience on the content of today's webinar through multiple choice questions. If you could select the response that applies most closely with you, that would really help us. So we will be launching the first polling question right now. It should be popping up on your screens. We just want to get an understanding of whether you've currently been using Benchling or have used Benchling in the past or not. Okay. So I think just a couple more seconds here. Great. So I think it's about a nice 50-50 split between those of you that have used Benchling before and are actually new to the platform. Thank you for that. So with that, I'll actually pass it over to Rashi to get started with the webinar.
Thank you, Ankita. Hope everyone's having a good morning or afternoon wherever you are, and you guys are staying safe in this current environment. So today we're going to be dividing the talk into 3 sections and really demonstrating how Benchling is bridging the gap between lab informatics and automation hardware. The first one is going to be an introduction to the Benchling platform for those of you who are new or unfamiliar with Benchling. The second part is going to focus on current trends and situation. There's been a lot of innovation and investment in lab hardware, but informatics appear to be lagging behind. And the third section would cover our new capability, Benchling for Lab Automation, how Benchling's connected suite can seamlessly integrate with lab instrumentation, including liquid handlers and analytical instrumentation. Benchling has been a trusted partner to life sciences leaders. As you can see here, we have a whole plethora of companies that we work with who range from working on biologics, cell and gene therapy, all the way to synthetic biology and agriculture. And our mission is really to help accelerate the pace of your life sciences R&D. So first, I'm going to start with a little bit of a Benchling introduction and talk about our different applications and how we can support you. So this slide is really to demonstrate that Benchling is a life sciences R&D cloud. It's a unified and biointelligent platform. And the main thing here is that your scientists will interact with one system and reduce any unnecessary work in transcribing info into multiple applications and places. As you can see on the screen here, you can interact with all the different applications that Benchling offers, including inventory, registry, notebook, molecular biology, all within the same screen. This really leads to the centralization, standardization, and interconnection of all of your R&D data in one place, whether that's your DNA sequences, to the physical location of your samples, to associated results and experimental contexts. Everything resides in one place so you can make decisions faster and gain new insights. So Benchling Life Sciences R&D is like a native cloud platform purpose-built for biologics R&D that really differentiates us from our traditional informatics players in the market that usually started off as small molecule players and then pivoted into biologics. Since Benchling was built from the ground up for biologics, we're easily able to model the complexity of biologics. Benchling is a fully unified and configurable platform, so you can track your biologics workflow end to end in real time with complete traceability. Whether it's your sequence, all the way to your mouse, your hybridoma, your lead, all the way to process development and getting ready for manufacturing, all of that data resides in one platform. Benchling is also a flexible platform that's easy to integrate and extend. We understand that you have a complex ecosystem with multiple databases and instruments to perform specialized actions. We're easily able to integrate with them. We also pride ourselves in implementing our systems in weeks and give your admins the power to adapt the platform to your evolving needs without any vendor involvement. What sets Benchling truly apart is our intuitive and modern user experience. We have tried to incorporate elements from your social media that you use in your daily life to Benchling, such as app mention, and also our modern interfaces has really led to an unprecedented user adoption and loyalty. That's truly one of the main things we hear from our customers all the time, that it's very easy to use and it's very modern, that really enables adoption for them and their scientists. So I'm going to talk about Benchling as a platform. Benchling is really divided into 4 types of applications. There are the productivity applications, samples tracking, process management, and analytics. The productivity applications consist of our molecular biology application as well as the notebook application. The idea here is that scientist has one window into the system, that's our notebook, where he or she can directly register samples in the registry or manage inventory without leaving that application. We also have high throughput molecular biology capabilities, including CRISPR, sequence design, batch cloning, and bulk assembly tools that's directly linked to the notebook. So you have all of your experimental context as well as your data and sequences in one place. We also have tools built within the system to ensure strict regulatory compliance, such as audit trails, version history, role-based permissions, and electronic signatures. So you never have to worry about whether your data is safe and it's protected. Our second type of application are the sample tracking applications. These really consist of the registry and the inventory. The registry can model complex biologics such as CAR-Ts, bispecifics, cell and gene therapy with clear linking and biointelligent relationships with its upstream and downstream counterparts. What that means is you have full visibility from the heavy chain, light chain, the sequence level, all the way to the final lead. And all of these links are clickable so you can find additional information or metadata associated with it, as well as the physical location, if it's in your freezer or in your liquid nitrogen tank. These apps can integrate directly with smart freezers, fridges, barcode scanners for standardization and a comprehensive view of your samples and reagents. Our third type of applications are process management applications that includes our requests and workflows. These apps really power effective and streamlined collaboration between your groups, whether that's core group and discovery group or an analytical group and another discovery group. This is really important to be able to kind of power that effective collaboration. Since all the requests now reside on one place, managers and executives are able to identify bottlenecks and improve resource allocation and productivity. With Benchling, now you have one platform that truly unifies discovery and development. So there's comprehensive transfer of experimental context as well as data between functions. Our fourth type of applications is actually our insight application that is officially launching in 2 days. So we're going to give you a sneak peek on it today. This application really enables scientists, managers, and executives to gain scientific as well as operational insights into your data. So you can ask questions about your experiment or across experiments, as well as any operational insights that you want to have, whether that's resource allocation or resource productivity. They will be able to now ask questions within Benchling using our user-level SQL queries. It will also enable users to visualize their data in graphics directly in Benchling and create dashboards and reports to accelerate decision-making. Now we're going to be moving on to our second polling question for the day. So here we really want to understand what scientific area that you or your lab is working in, so we know how to support you better. Just a few more seconds. Great. That's amazing. We actually have a really good mix of customers all the way from antibody, gene therapy to agriculture and synthetic biology. So yeah, I hope the rest of this webinar will be very informative for you. All right, so now we're going to be changing gears to the next part of our talk, where we really want to discuss some current trends affecting R&D organizations that really propelled us to expand our offering and include and really do this new capability, Benchling for lab automation. So we've really been seeing that lab automation has become a critical part of life sciences R&D. We've been seeing this shift for almost a decade now. There's several market dynamics at play, whether that's emergence of new complex drug modalities, increased competition, or really a desire to reduce costs. This has contributed to, I'd say, three main trends. There's increased throughput requirements. So i.e. customers really want to be screening a lot more samples early to improve their probability of success. The second is they want to be able to increase their testing. So not just test a lot of samples, but also test a lot of samples with different kind of assays, so they can really fail fast and fail early. That's really a mantra we constantly see with our customers. As a result of that, they're also now, you guys are generating a substantial amount of fragmented and highly complex data, much more than we've ever done before. But as you're starting to generate this data, you're starting to test more samples early, we believe that companies are still not able to maximize their benefits from their automation investments. This is because lab informatics remain disconnected from their instrumentation and hardware. Traditional LIMS systems are very good for fully established process or one process, but it's often rigid and hard-coded for a specific process and hard to adapt to a new workflow. Specialized solutions, whether homegrown or commercial, are often siloed for a specific group or a few groups and often lack the upstream or downstream context about the samples. This creates a bottleneck for data aggregation and end-to-end traceability that's really important. We often hear our customers say they always want to keep the sample chain of custody intact throughout all the process. But in order to really aggregate that kind of data, it's often hard if they live in different systems. And we believe that really this impedes your pace of research. And that's why we built Benchling for lab automation, to really help you harness the full potential of your lab hardware. So Benchling for lab automation, it's truly an out-of-box integration with liquid handlers and analytical instrumentation. The idea here is that the scientist interacts with a simple run object in the notebook, and all of the underlying actions, whether that's registry, registering new samples, managing your inventory, and requests are automatically performed for them. So let's dive into a little bit more detail. And today we're going to be giving you a live demo about the product as well. And Jun's going to be doing that after me. So I wanted to first give a little bit of a context on the product. So there are really two aspects of the capability of the Benchling for Lab Automation capability. It's generating run instructions for your robot and ingesting output from either your liquid handler or your analytical instrumentation. The main thing to really understand here is, if you see the cartoon on the right, is Benchling holds the entire context of your sample, whether that's including sequence, the plasmid info, and all of this is, and where it's located. And all of this is really leveraged to generate run instructions based on scientist-driven input samples and parameters. The other main thing is that these run instructions are actually generated using point and click with no custom code. So if it needs to be modified, if your process needs to be modified, this can be done within a few minutes by the customer admin directly. The run instructions are really CSV files, and this is usually given to the robot. So the robot will use the files along with its own control software to perform the actions. And once it performs the actions, it produces an output. The second part of this capability that Benchling really offers is being able to ingest the output that's coming from liquid handlers as well as analytical instrumentation. The configuration here is really easy, and it's done with one API endpoint. What that means is whether you're creating a results table, whether you're doing anything to the registry, the inventory, or just taking in data, all of that is done with one API endpoint, and it leads to a number of actions. These actions include, you can create a results table in your notebook. So you're taking analytical data. We're going to be demonstrating how Jun will be taking qPCR data and putting that results table in the notebook. Whether that's new samples in the registry, let's say you're creating new samples or updating samples, all of that will be automatically performed in the registry. If you're using some kind of reagents or samples that's in your inventory, those volumes will be automatically decremented. All of that info will be updated with using this one API endpoint. So all of the underlying actions Benchling will take care of it once it ingests the output. So before we jump into the live demo, I want to highlight 3 aspects that makes this capability truly unique compared to other systems. The number one is it's built on top of a unified platform. So all relevant info is integrated automatically into Benchling applications without user intervention. That really reduces the time spent and decreases the probability of introducing error during transcription. We often hear this, that when customers are when they get the results from these different analytical instruments or liquid handlers, they have to manipulate these Excel files or CSV files before importing it into a LIMS system. That may introduce errors. We're taking that middle step completely out of the picture, where we're ingesting it directly and updating all the applications that need to be without any user intervention. The second one is critical, is that Benchling really has all samples are directly linked to their upstream and downstream biological counterparts, so they have rich sample and process intelligence. What that means is you can access info from one window on the mouse, your tissue, your hybridoma, your subclone, and all the associated results from one window. And Jun will be kind of showing you that, where the lineage is kind of built up as you're creating new samples or as you're updating new entities. The third aspect that truly makes Benchling for Lab Automation unique is that we're not hard-coded, which gives you, the users, the flexibility to modify to a new process by admin very quickly. Now I wanted to actually do another quick polling question while Jun sets up and gives you a live demo.
All right, thank you, Rashi. So I think we'll let this go for just a few more seconds. All right, maybe 5 seconds more. Great. Thank you everyone for your answers. It's helpful to see what kind of liquid handlers you guys are using. It looks like there's a good portion of you that are using Hamilton instruments, so that's great. For those of you who just logged in and missed the introduction, my name is Gene Crisaldo. I'm a scientific solutions consultant with Benchling. I'm a molecular biologist by training, and my role with the company is to evaluate scientific and technical fit with our product. So we have some pretty exciting things to show you, and I'm really looking forward to showing you this. What I like to do when I go through the platform is I sort of like to mock an example use case. And I sort of want to mimic a day in the life of someone who would actually be using Benchling for a specific purpose rather than sort of just broad capability showing. So the use case I'm going to be mimicking, and while this use case is pretty specific to this one scientific area, know that a wide variety, our product applies to a wide variety of different scientific modalities. So I'm going to be basically walking through a strain engineering use case where either a group or a specific person on my team designed some vectors in silico with some genes that they were targeting. They assemble it, they amplify it, and then they choose some bacterial host that they want to use. They make those cells competent to take up the vectors, they culture it, and then based on some OD threshold, we'll perform some hit picking, and then we'll verify the sequence uptake via NGS. So in that process, we'll use our liquid handlers to do the hit picking, do the library prep, specifically the adapter ligation, and then also do the pooling step prior to actually loading it onto the sequencer. One thing to note about this process that you see here is that this entire process could actually be captured and modeled and supported entirely within Benchling. Particularly these first 4 steps that you see here, you can imagine that our molecular biology suite will support the bulk design of our vectors as well as modeling the sequences. Our notebook is used to record the experiments that take place, such as the assembly and the transformation or the culturing. Our registry actually models all of our different samples that are relevant to me or my team, such as the different strains, whether they were transformed, the media, the buffers that are associated with it, and as well as the inventory for modeling all of those components physically in the lab. And to carry forward with this process, this is where we're going to use our notebook to carry out the hit picking, the adapter evaluation, and the library pooling. But particularly, you'll see how Benchling for Lab Automation is used in the context of the notebook. So your scientists or your lab technicians, as they carry out these experiments, they're familiar with the notebook, they're used to recording experiments in it. So we sort of want to keep that interface and have Benchling update the rest of the platform that interfaces with your experiments, but also interfaces with your liquid handlers here. So what you'll see when I get into the platform is this object called a run. And I'll explain what that means in a moment, and you'll see that further as I walk through the platform. But within the body of your notebook entry, you'll see this object called a run. And basically what this run does is it allows you to configure exactly what you want represented in your input CSV file. So particularly, you could either choose your plates, you could choose your sample IDs, you could configure the volume, you could probably have a drop-down of method names, basically whatever you want. And by choosing those fields or configuring what you want, you simply just hit a button and you'll generate this unique CSV file that could be brought to your liquid handler controller software. And then after your liquid handler processes that input file, most liquid handlers will spit out some type of output file that records all of the steps that took place. You could also bring that output file back into that same run object. And the reason why we do that is so Benchling could actually reconcile exactly what happened. So it'll either create new plates, it'll transfer the samples, it'll record the volumes. If your liquid handler run produced a new entity, for example, if your liquid handler was performing a DNA extraction method, Benchling will record new DNA entities if you wanted to record that. So within that same run object, it'll generate the input file as well as process all of the output files that were produced. So with that said, let's go ahead and jump into the platform. I do want to reiterate that as you're seeing me walk through this, you might have questions that come up. Please use that Q&A box on the bottom to please submit any questions. I probably won't be able to answer right away in real time, but towards the end when we do Q&A, we could go ahead and address some of those questions. I did see a question during Rashi's presentation that someone asked what's the difference between the registry and the inventory. So I'll go ahead and actually walk through that. So there's a good chunk of you that have not seen Benchling before or have not used it. So I just want to give a quick orientation to what the platform looks like before I actually show lab automation. For those of you who have used it, this will be just a quick review of things that you're already familiar with. So I'm currently logged into the platform, and here I have a notebook entry opened up where I'm performing my strain screening experiment. If I want to access different parts of the platform, I could do that via this navigational toolbar on the left. So you can see I could access my projects, I could do a global search, I could access my registry, and I could access my inventory. So really quick, the registry is used for modeling all of the components that are relevant to your lab. So think cell lines, think strains, think plasmids, think protein sequences, DNA sequences, mice, instruments, basically anything that's important to your lab, you can model in the registry. The inventory is for modeling all of those components in a physical manner. So rather than modeling the actual biological entities, you're modeling fridges, freezers, racks, shelves, boxes, shelves, plates, and whatnot. What's neat here from the notebook, you'll see right off the bat that in my notebook body, I have these blue hyperlinks here. As Rashi mentioned before, we want to bring in some tools that you're probably used to using every day in your personal life here into Benchling to make that a bit more intuitive. So these blue hyperlinks are essentially accomplished by typing in the @ symbol, and you could actually link or reference any other thing that's within Benchling into this notebook experiment. So for example, as part of my experiment here, I'm referencing these 3 plates that are filled with transformed strains that I need to screen. So you can see here, I'm actually going to reference my colleague, Abigail here. And what's neat here is when I reference her, I could go ahead and send her a message. And she would actually get notified via email. And if she had the appropriate permissions, she could go ahead and log into this notebook entry, start making edits, as well as see this message here for me. So if I wanted to see more context via these links, for example, I want to just quickly check what my strains here look like. I could go ahead and click into one of these plates and then immediately see more context here. I could see the barcode of my plate, I could see the name, I could see the location it's in, depending on how I'm modeling the unique locations in my lab or in my organization. And if I look here, I could immediately see the unique volumes of my samples, as well as I could see links to my samples themselves as well. So if I wanted to see a bit more context about this particular strain, I could go ahead and click into that. And then you can see Benchling aggregates all of the context in one place. And this is the same across all of my different sample types. So you can see here for my strain, I see some pretty consistent pieces of information, such as the author, the project it's associated with, as well as standard metadata fields here that I want to capture against my strain. I do want to highlight here that these fields are configurable. So you could configure this to map to whatever kinds of metadata are important for your sample types. So in this particular example for my strains, I just want to keep track of the vector that I've used to transform it, as well as the date that was transformed. I could probably add more things here, maybe buffer information, maybe lot ID information, maybe vendor information. Essentially, whatever is relevant to you, again, you could add those fields here and standardize it that way. So to show you here how Benchling is linking the context, so we have the strain information. I could also see under this results tab, I could see some assay data that was generated against it, such as plate reader information, and I could see who generated it, as well as the original source. So I could go back and see historical legacy data all in one place without necessarily having to look through, lots of different paper notebooks or look through a handful of different Excel spreadsheets. And additionally, I could go down to the sequence information. If I wanted to see information about this vector, I could go ahead and just jump into the vector. And then from here, I'm jumping into my molecular biology suite where I could perform some more sequence analysis and have access to these tools. Rashi mentioned in her talk that Benchling does the work using what we call biointelligent links, where Benchling on the back end does some of the work of, again, linking all of these things together. So for example, in my strain here, if I go down here to relevant items, Benchling will automatically link all of the other entities that are referencing or have come from this particular strain. In this particular example, I have my notebook entry that I've just opened up prior. automatically down here under relevant items. So I could go ahead and jump back into that and continue with my work here. So as part of my strain screening experiment, the first thing that I'm going to do is I'm actually going to use my liquid handler to do some hit picking here. So I've described what a run object is, and we're going to see that in action. So I have an example of a run object here that's particularly performing my strain hit picking step, as well as isolating the DNA from my strains. So you can imagine that on my liquid handler, I went ahead and I programmed a method that does the cherry picking and then also carries out the DNA isolation method by pipetting the reagents if it was bead-based or column-based, whatever your method is. So I've configured this run where my user or myself just decides what strains I want to pipette, the volume, as well as the final DNA destination plate. So to do my hit picking, I could actually apply a threshold within Benchling. So I'm going to go ahead. I've went ahead and created a dashboard here where I took those 3 strain plates and I'm just graphing all of the OD values from the plate reader data set here. So I have about 288 strains here. I want to go ahead and apply a threshold to apply to my strain hit picking. And just really quickly, you could see that I'm filtering all of my different strains by the different vectors that transformed it. So I'm going to go ahead and apply a threshold here. And let's say I only want the strains that meet this threshold. And you can see it filters down even further. And then, luckily for me, I have roughly 96 strains that got filtered down. So what I could do now is I could take this list and actually apply it to my run here. So I'm going to go ahead and create my input file that will perform the cherry picking hit list here. So I'm going to go ahead and create this run. I'm going to go ahead and add samples from that cherry picked hit list. Going to choose the volume of strain that I want pipetted, as well as the final plate that will have my DNA. And I'm going to draw or choose this plate from another table in my notebook entry where I've labeled just an empty hit-picked DNA plate. So I'm going to go ahead and just save those fields there. And then you can see down here is where I could go ahead and generate my input CSV file. So I'm going to go ahead and generate that. And you can see a unique identifier is generated to the CSV file, as well as with a unique timestamp. So I'm going to go ahead and open this guy up. I know someone asked if there's an example CSV file that I could show you. So I'll go ahead and open that up. And the CSV file essentially looks like this, where we have columns here, and then we have the rows. I do want to highlight here that the columns that you see here is completely configurable. We're completely agnostic to any platform. So as part of our implementation with you, and also with continued configuration flexibility that you will have, you could modify how these columns are generated to match the liquid handler or to match the analytical instrument that you currently have. So what you see here is just a broad example that I've put together, but just know that we're very flexible and not all liquid handlers will take the same format. So you could configure this to match exactly the formatting that your liquid handler can take. So again, via either our APIs or done manually, I could take this CSV file to my liquid handler. Let's say, obviously I can't show you this, but let's pretend that the liquid handler has performed the strain hit picking method as well as the DNA isolation method, and it spit out an output file. I can now bring that output file back into Benchling. So again, via done manually, what you'll see here, or using our APIs, we could bring that output file back here. So I'll go ahead and just import the same file here just for the sake of an example. And then I'm going to go ahead and process that. So now what Benchling is doing is it's reconciling the 2 steps that occurred here. And now you can see the processing is done. I could go into my plate that was previously empty, and Benchling automatically went in and filled in the volumes that I've described. As well as created these new DNA isolate samples. And if I go into one of these DNA isolate samples, you can see Benchling automatically maintains the context here, such as the original strain that the DNA was isolated from, and including the source vector that is denoting this DNA as well. All right, so moving forward in my experiment, so now that I have my isolated DNA, I could go ahead and move forward with my library prep process. So here I've designed another run object, and this time I've designed the fields to reference the new DNA plate that I've just created, a field for me to select my index plates, as well as determining the volume of my DNA and the volume of my index. So let's go ahead and I'll edit those fields here. So I'm going to go ahead and just select my new DNA plate that was just created in the previous run. I'll go ahead and select my index plate. Let's say I'm just choosing some indices I ordered from IDT, my volume of my DNA, my volume of my index, and then lastly, my final library plate that all of this will be pipetted into in the end. All right, so I'm going to go ahead and save those fields here. And then in the same process, you can see all I did was just choose my source plates and my destination plate and the volumes. I'm going to go ahead and just generate that input file. And I'm going to go ahead and open that up. I'm not going to show you it because it just looks more or less exactly the same as the previous example that I've shown you. But again, just want to reiterate that you can configure that to match whatever liquid handler platform that you have. So in the same vein, I take this to the liquid handler. The liquid handler does the library prep process, spits out an output file, and I can now drag that output file back into Benchling. I could go ahead and process what happened. So Benchling is reconciling those two steps together. And then as soon as it's done processing, you'll see this nice little green check mark here. I could go ahead and go into this plate, again, that was previously empty. And I have my new volumes here, as well as my new library entities. And again, in the same vein, if I go into one of these library samples here, you can see Benchling is still automatically maintaining the context, and I didn't have to enter in any of these fields. Benchling is doing that for me. So it's maintaining the original DNA isolate that created this library, the index that was used. Obviously, if I'm using a paired indices for each one, I would have probably both of those recorded here. The original strain that this library was derived from, as well as the original vector that transformed this strain. Additionally to using these runs to generate input files for liquid handlers, as well as processing output files, you could also use runs to process output files from analytical instruments as well. So in this particular example, I've designed a run here to just basically take an output file, let's say from something like a Quant Studio or some other qPCR machine here. So I'm going to go ahead and just upload some results. Again, you're seeing me do this manually, but if you wanted to use our APIs, this can be done automatically as well. So I'm going to go ahead and just drag in, let's say I had some PCR data that came off of an instrument. I'm going to go ahead and just bring that in here. And you can see Benchling is mapping the columns of the spreadsheet to just make sure it's correct. So I'm going to go ahead and import that. And then you can see here with my new libraries that I just created, I now have recorded some arbitrary qPCR data as well. And what's neat here about recording it like this is that in the future or down the line, let's say if I were to access this library sample, I could go ahead and go into that. And then Benchling automatically records that qPCR data under this results tab. So all the data is automatically aggregated in one place and attached to each sample. So I don't necessarily have to look for this notebook entry. I could just look for this particular sample and then see all of the historical assay data that was generated against it. All right, so then moving along here, as part of my final step, assuming that the libraries pass QC, I can now have this final run object here that will generate the input instructions for pooling all of my libraries together into a tube so that I could load it onto, let's say, my MySeq. So I'm going to go ahead and create a run here. I'm going to reference my library plate that was created in my previous run object. I could determine the volume of library that I want in the final pool, as well as selecting the final tube that will be generated here. All right. And in fact, I actually need to go ahead and create this tube here. So let me go ahead and create an empty tube. So you'll see a quick peek into the inventory here. So I have a box of my pools here. So let me go ahead and just create a new container. And I'll just call this MySeq pool 4. and it'll just be right there. Just gonna go ahead and create that. So that will be the tube that I will pipette my libraries into. So you can see now that I've created that tube, I could go ahead and just reference it here. I'll go ahead and save those fields and then generate this input file. So as one final step, let's say I take this to the liquid handler. Liquid handler pulls all of my libraries together, spits out an output file. I could bring that output file back. Go ahead and process what occurred here. And then in the same vein that Benchling created, the DNA isolates created the libraries. It has now created this pool entity. And if I go into this pool entity, you can see that it keeps track of all of my libraries automatically in one place here. All right, so you can see that with Benchling for Lab Automation, Benchling will automatically update the context of your samples in the registry, as well as automatically move them in our inventory. And you can see just with just a couple of clicks and not really having to keep track or type in anything, Benchling will generate all the files and maintain all of that context all in one place for me. So with that, before we actually go into Q&A, I could see that there's a lot of questions that were popping up. I'm going to hand it back over to Rashi, but we also have just another polling question that we want to ask you, and basically want to find out what is your average sample throughput.
Perfect. Thank you, Jun. I think just a few more seconds. Perfect. So it looks like most of you are between about 10,000 or less samples per week. That's a very sweet spot for Benchling. So we'd love to definitely talk to you more after this webinar. And I said we do have a Q&A session, but before we get there, just a quick case study that I want to talk about. So we did roll out the Benchling for Lab Automation capability with an industrial biotech leader. This was as part of our Lighthouse or beta program, and their main objective was to really accelerate and streamline the enzyme discovery process, because they make enzymes for industrial applications such as laundry detergents, increasing the nutritional value of food, processing textiles in a more eco-friendly way, to name a few. This was for an NGS group that followed the same workflow that June showed. And the main thing was that they had a company directive to be able to decrease their campaign. They were running about 5,000 samples per campaign, and it would take them about 2 weeks to run that. There was a company directive to decrease that time to 1 week. And in order to really run this campaign and run this NGS workflow, they had multiple homegrown systems that handle each section of the workflow. There were about 4 to 5 systems that didn't talk to one another, and they generated about 30 to 40 Excel files per campaign. Their campaign took about 2 weeks, as I mentioned, and there was a company directive to reduce that. As they were running multiple campaigns at one time, handling the multiple Excel files and systems became a true bottleneck for them for decision making. In addition to that, they were also seeing that the naming of their oligo preps were not standardized. That led to a lot of redundancies and time wasted, because either they couldn't find it, it was sitting in an Excel sheet, or they had to reorder it, but it was sitting in a freezer somewhere, but they didn't have access to it because they didn't have that context. Another main challenge that they faced was that all the data output files that were coming from their instruments. So the 3 main instruments they used in their workflow was the Echo, the Biomech, as well as a colony picker. They had to be manually manipulated and uploaded into one of these disparate systems that was often error prone for them. So to really help them, we implemented the entire Benchling solution, which included the notebook, the molecular biology, the registry, inventory, the requests, as well as Benchling for Lab Automation. And what really helped them was that now they were able to replace 4 to 5 disparate standalone systems by a single solution. And as Jun mentioned, they were able to update their registry, their inventory, as well as have results in one centralized location. They also noticed a 30% increase in scientist productivity because now there were improved capture because all of this was manually being captured, automatically being captured, and there was no duplication because previously they had to add some of the context into each system multiple times. They also noticed a 50% time reduced per campaign, which was as part of their company directive, because they were able to directly integrate with these instruments, as I mentioned, the biomech and the echo and the colony picker, and really have this one end-to-end solution that gave them complete traceability. I do want to mention here also that a few of you had these questions as well, and we tried to kind of gauge with one of our polling questions. Benchling for lab automation can easily integrate with almost any laboratory robotics and instrumentation. We have partnered with companies like Hamilton, TCAN, and PAA to demonstrate how easy it is to kind of plug and play Benchling software solution to work hand in hand with your hardware investments. And we really hope to expand these partnerships to more vendors in the future. However, we are still vendor agnostic and completely configurable. So this won't, just because we're mentioning these 3 plug and play partners, we are able to integrate with all instrumentation as long as they have a CSV input and output. So to end today's talk, I do want to highlight the Benchling difference and why Benchling has truly become indispensable for next generation life sciences R&D. We really tried to unify hardware and informatics. And as we mentioned at the beginning, the trend where informatics we believe was kind of lagging behind hardware, we're really trying to close that gap and give you that end-to-end traceability so you can have all of that context upstream and downstream of your samples. Since all of your information, whether that's results as well as experimental context, resides in one place, you're able to really accelerate your speed to milestone and have access to data-driven insights, whether that's experimental or operational. And so we'd like you to start your transformation with Benchling. We're going to open the floor up for new questions. Just quickly, new users, you can request a demo using this link. And existing users, please contact your Benchling customer success rep, or you can definitely ask us questions about it today as well.
Great. As Rashi mentioned, we'll be moving into the Q&A session once this poll is closed. We have a lot of great questions that have been streaming in throughout the webinar. Great. So I think just a couple more seconds here. Fantastic. Thank you all for that feedback. It's very helpful for us as we continue to develop our webinar series. So jumping into the questions, Jun, maybe this is a good question for you. Do we need the entire platform to get Benchling for lab automation capability?
Yeah, it's a great question. I think as you saw in the demonstration, how the lab automation run object was automatically updating the registry and the inventory, you would find the most value of using lab automation with the registry and the inventory. I think part of the value with it is Benchling provides all of the context. So we want to be able to maintain that context to its fullest extent.
Fantastic. There were several questions on how the integration between Benchling and liquid handlers or instrumentation works. Can one of you quickly summarize how that integration works? And specifically, can it be used with multiple file formats? And can it be used with maybe existing e-notebooks that can be connected to this?
Yeah, I could take that. So as far as, I think there are 3 parts to that question. So the first part about how specifically do we integrate with other liquid handlers or instruments, it's pretty variable. So as you saw in my example, one common integration, and this is actually less of an integration and just more of a manual process, you could see that that unique CSV file was generated. You can imagine that if you had like a shared network drive or like a USB drive, your scientists or your research associates could just drag that CSV file into your liquid handler controller software, and in that way it's integrated. And same in the reverse direction with whatever output file was generated from the liquid handler. Other integrations actually use our suite of APIs. So you could imagine using our APIs, if you had some shared network drive that's connected to your liquid handler, you could have Benchling push that file into the shared network drive, and the liquid handler would read that shared network drive with any updates, with any files that populated it, and then just bring that file into Benchling. And then same again in the reverse direction. If the liquid handler spit out an output file that goes into shared network drive, there's a Benchling script that looks at that shared network drive every so often or every interval, and then just updates Benchling with a net run with any file that's updated there. As far as different file types, currently we do support CSV formats, but we are constantly improving the product. We understand that other platforms probably support other file types such as text formats. So that is something that is constantly being improved, or even .xml. So currently it is CSV, but the product is continually being developed to support more use cases. And as far as integrations with other laboratory notebooks, in general, we do support integrations with other types of software beyond even just lab notebooks. But particularly for Benchling for Lab Automation, we do think that you find the most value with it with using our notebook because of how easily connected it is to the rest of our platform.
Great. The next question is about outsourced testing. Can Benchling for Lab Automation be used for uploading any outsourced testing or outsourced files?
Yeah, no, that's a great question. As you can see, it's purely just up to you on how you want to bring in that file type or that file of data into Benchling. So whether that's, you decide to buy licenses for whatever is outside of your company or your organization, or if you just have a system in place for you to receive the files that they produce. The main thing to figure out is how do you get those files that have that data, and how do you decide you want to bring that into Benchling? So short answer is yes, but definitely on a case-by-case basis, we would have to discuss what your particular context is so we can figure out how to best support that.
Fantastic. The next question is in regards, I think it's to the broader Benchling platform. How do you integrate either through APIs or links with Google Sheets or any G Suite applications? Can you import a Google Sheet?
Yeah, good question. So I didn't show this in the platform, but we do have, there was an icon in a navigational toolbar called external files, and you can actually just directly link up Google accounts. So you can bring in Google Documents or Google Spreadsheets directly into Benchling or reference them. Depending on what you want to do, we would advise on the best way to sort of bring that data in. So it could be as simple as just copying and pasting from a spreadsheet, or it could be as simple as just linking out to a specific link.
Great. There was another question here in regards to the Benchling platform and how applicable it is to working in a regulated environment.
Yeah, great question. Rashi, do you want to take that?
Sure. So as I mentioned briefly, that we do have tools to support 21 CFR Part 11 compliance, whether that's role-based user access, very controlled access, electronic signatures, very comprehensive audit trails, version history. But we are also continually improving on that by adding more GXP capability, and we hope to be launching that very soon.
Great. Now coming back to the Benchling for Lab Automation products specifically, there were a few questions in regards to how the input file was created. Can one of you speak more to our runs feature and how that input file is generated through the Benchling platform?
Yeah, definitely. So as far as on the back end, when you're designing that run, if you're familiar with Benchling terminology, we define things like a run or sample types as schemas. And within that schema, you define different types of metadata. And that could be whatever you want. So particularly for run schemas, you're essentially defining in the back end what type of columns you are mapping out. So you define the column headers. And then within each cell underneath that column header, you're defining what type of object will go in underneath that column header. So whether that's a sample ID, whether that's a plate ID, whether that's a volume transfer amount, whether that's an arbitrary method name, essentially whatever you would want. So you're essentially defining what fields your user needs to enter. So basically what they see in the UI. And you're also defining what columns are filled or populated based on those fields that are entered.
And I'll just add that that's truly, as Jin said, it's totally configurable. And that's what makes us kind of vendor agnostic. So irrespective of what liquid handler you have, whether that's as Hamilton, TCAN, Beckman, right? Like we're able to do that because we can configure those columns to match how your liquid handler will read that input file.
Great, that's really helpful. That also, I think, answers the next question I had here in regards to can the data be rearranged in the CSV file, and can that be done in like the rows and column formats within Benchling?
I mean, I'll just add one more thing, that the idea is that we don't want you to be manipulating the CSV file, right, to kind of reduce any data integrity possible issues, which is why if you can configure the run schema to have those exact rows or columns that you need, that would remove that step altogether.
Fantastic. There were several questions about whether the product works better with specific liquid handlers or instrumentation over others. Can one of you answer that?
Yeah, go for it, Rashi.
Yeah. So the answer for that is no. As I said, we do have plug and play partners. However, there is no preferential vendors that we believe like it works better than with others, as long as the instrument is able to take a CSV input and produce a CSV output. It should be easy to integrate with Benchling.
Fantastic. So I think we're just at the top of the hour right now. There are still a few questions that we haven't answered, but we will circle back over email. So thank you all for submitting questions and being such an engaged audience. Thank you, Jun and Rashi, for taking us through this content. We will be sharing a recording of the webinar within the next 2 business days or so. If you would like any additional information, please feel free to visit our website at benchling.com and request a demo, and we can speak to you more about how our platform and this product can be leveraged for your specific scientific use case. Thank you all again.